So what is DNA cloning? DNA cloning is a technique for amplifying and or, which means it is optional. You could either only be amplifying the DNA and you could also be expressing your DNA fragments. And there are two basic methods of DNA cloning. One that is known as the in vivo or cell-based method, which we're going to discuss in more details in current lecture. And then the other method of amplifying a piece of DNA and cloning DNA is what is known as the in vitro method or the PCR or polymerase chain reaction based method. In this case, you can only amplify the piece of DNA. You cannot really express it. So the expression does not happen here. Only the amplification happens in vitro or PCR based method of DNA cloning or DNA amplification. So as an activity, I want you to look for the meaning and application of the following terms: in vivo, in vitro, and ex vivo. So let us now talk about the steps in cell-based cloning, some of which we've already studied and some of which we will be studying today. So first, of course, is preparation of insert. Insert is your DNA of interest, the one that you want to actually clone and multiply and or express. And in previous lectures, if you remember, I told you that most commonly it is the sticky end ligation that is preferred. And if the insert does not have sticky ends or staggered ends, then ends can be generated by either adding the linker or the adapter or homopolymer tailing. So here is your insert, and in this case, we have added an adapter at this end and at the other end so that this has now sticky ends. The second important point here is the preparation of the vector. So you have to first make a choice of vector. This choice of vector will depend on the size of the insert, because depending on which vector you select, there is a limitation to the maximum size that can be tolerated. And we'll see this in more details as we move into the lectures in vectors. And then, of course, the other important point here is the objective of the experiment. If the objective of the experiment is only to amplify the DNA, a normal vector would do for you. If the objective of the experiment is to also express the protein, then of course you have to take expression vectors. And most vectors come as circular DNA molecule, which has to be opened up or linearized so as to ligate it to the insert. So that process of linearization also has to be taken care of. And the linearization of the vector takes place by digestion with restriction enzymes. And in unit 1, we have done restriction enzymes in details. So we'll not go into so many details of that for now. So here you are. So this is your plasmid vector, closed covalent circular DNA, which is opened by restriction in the nuclease at a certain point. And this is the point where your insert is now going to be inserted into this plasmid vector, and the gaps are resealed, finally giving you again a circular DNA molecule, which includes the DNA of the plasmid as also your DNA of interest in a single DNA molecule. So this is your whole process, and the next, of course, is the process by which the two molecules become single covalent DNA molecule is what is known as ligation. So ligation is where you have your linearized vector, you have your DNA of interest or insert, and you put them together and use ligase enzymes so that they now form a single DNA molecule. And then beyond this, we have the steps of transformation, which is basically to insert this recombinant DNA molecule into a host cell where it can autonomously multiply. And when the host cell multiplies, it also multiplies with that. So this is the process of transformation, and transformation itself has several steps and several ways in which you can transform the host cell. So this is these include heat shock, electroporation, and gene gun. And we'll discuss each of them in more details as we move along. After the transformation treatment, you have to allow the cells to recover, and they have to be grown in rich medium so that the cells come to a normal stage. And then beyond that, you can grow them in specific medium, specifically with antibiotics, so as to ensure that you are able to select only the cells that are transformed. We'll discuss this step also in more details. And then finally, of the cells that come up upon the selection criteria, you have to basically isolate the cells that are truly recombinant from the cells that are not recombinant. So that is what is known as selection, and we'll also discuss selection in more details as we move along. So this process, this part of transformation, recovery, and selection is what most of the lecture is going to deal with after this. Next, let's talk of the key players that are involved in cell-based cloning. So the key players include vector, insert, and host cell. So let's see the vector here. So vector most commonly will be a circular covalent DNA. We take the example of plasmid for simplicity. A vector is required to carry DNA fragment of interest into the host cell. And the idea is that because your DNA of interest or insert is ligated into the vector, and as the vector moves into the host cell, and because the vector also has the property of autonomous replication, so as the vector replicates, your DNA of interest is also going to replicate. And then, of course, as the cell divides and further grows in numbers, the number of copies of your initial DNA that you put into the vector also multiplies. So this is the whole idea of DNA cloning or DNA amplification in live cells or in vivo conditions, right? Then you have the other player, that is the insert, and I've already told you insert is basically a piece of DNA. This could be genic, this could be non-genic. You may be cloning a piece of DNA for expression of the protein. You may also be cloning a piece of DNA just to identify what sequence it carries. So depending on the experimental objective, this insert or DNA of interest could be genic or non-genic, and this then is ligated to the vector to form a recombinant vector, which then enters into the host cell. So insert is the DNA of interest to be cloned for multiplication or expression. And then, of course, you have the host cell. This is the cell into which your recombinant plasmid that carries the insert now is going to be inserted. And in the host cell, this recombinant plasmid is going to multiply autonomously, in the sense independent of the cell multiplication. And also, as the cell divides, you will have further multiplications resulting in exponential amplification of your piece of initial DNA that you've given into the vector. So this is your whole idea of the three key players. You have vector, you have insert, and the host cell. So vector is your transport vehicle, insert is your DNA of interest, and host cell is basically the cell in which the actual cloning or multiplication of DNA would happen, right? So this is the whole idea of the three key players in the game. Now, as an activity, I want to ask you which class of restriction enzymes are preferred for DNA cloning and why. And the answer to this lies in my previous lectures. So in case you're not able to answer this, you have to go back to my previous lectures and look for the lectures on restriction enzymes, right? That is lecture number 1. So the first step is linearization or opening of the vector. And in this case here, I show you a plasmid that is opened up by restriction endonucleases. You know, in this case, specifically, we use restriction endonucleases of class 2 because class 2 restriction endonucleases can cut uniquely and within a recognition sequence. So therefore here, this vector opens up or it gets linearized. And now in the next step, what you do is what is known as ligation. Ligation means where you are going to generate the recombinant vector. So here is a linearized vector. You add your DNA of interest or insert, and you use ligase. After ligation has happened, some cells would get the recombinant plasmid where you have the plasmid DNA and the DNA of interest coming together and forming a single covalent DNA molecule. And in some cases, the plasmid will self-ligate and form what is known as a self-ligated non-recombinant plasmid. And these two will be parts of a ligation mix, which will be taken forward for the next steps of the reaction. So then the next step in this process is what is known as transformation. And this is a key step in the process of DNA amplification, because ultimately the DNA is going to amplify only in the host cell. So transformation is a process by which an organism acquires exogenous DNA, the vector here, right? So in this case, we're talking about the vector, which is a recombined vector that we're looking for here. So if you see here, this is your recombinant plasmid, this is a non-recombinant plasmid, this is actually your ligation mix. To it, you add your host cells, and then, of course, over time, by certain treatment, which include electroporation or heat shock, what happens is your plasmid is able to enter into the host cell and form 3 types of cells, 1, 2, and 3. So this one here is a cell where a recombinant plasmid has entered. So this is a transformant which has a recombinant plasmid, and this is the one we are looking for actually, right? Then in the other cases, what could happen is that your non-recombinant plasmid, which is a self-ligated plasmid, gets into the host cell. So this is your transformant, but here the plasmid is non-recombinant, right? And then in the third case, where there is no transformation happening, which means that this cell would not have the properties conferred by the plasmid here. So this is your third cell, which is basically your non-transformant. These two are your transformants. This is a non-transformant. So here you are. This is a recombinant transformant. This is a non-recombinant transformant, and this one, third one, is your non-transformant. And then, of course, there are methods of screening out and following out and identifying cells that are truly recombinant. And then, of course, they can be taken forward for further cloning or expression experiment. Let's have a look at the methods of transformation in more details now. So here you are. The first important point in transformation is that the cells that you use for transformation, the host cells that are used for transformation, are not the normal host cells. They have to be in a state that is known as competent to be able to accept an exogenous DNA. Right? So this is your, again, the ligation mix. You have the recombinant plasmid, you have the non-recombinant plasmid. You have the host cell that is converted into a competent state so that it is able to, and it is amenable to accepting foreign DNA during the process of electroporation or heat shock. So this is finally what you get. Ideally, this is what you want to have alone, but of course you get two other types of cells as I showed you in the previous slide. So the cells that are used for transformation are special in the sense that, number one, these cells lack the vector. So the host cells that are used for transformation would not have the vector to begin with. So these are the cells that also do not have the properties that are conferred by the vector, right? So if the vector has certain resistance gene, so these cells would be susceptible to that particular antibiotic in the absence of the vector here. And second important property, which is also very significant, is the fact that they undergo special treatment to ensure that they are more receptive to intaking of foreign DNA, and such cells are known as competent cells. So naturally, cells may not be competent, but they can be converted into competent state by chemical treatment, and we'll see how this is done in the coming slides. So here is your non-competent cell, and by treatment, either chemically or by electrical shock or by other methods, it can be made into a competent cell where the outer membrane of the cell is now more porous, and because it is porous, it is going to allow for insertion of the foreign DNA molecule into the host cell. So this is the process we're talking about, where a non-competent cell can be converted into a competent cell by chemical treatment or by electrical treatment. Most cells are not naturally competent, but they can be converted into competent state. And they need special treatment to make them permeable to foreign DNA, that is, to make them competent. Some cells are naturally competent. These include Streptococcus pneumoniae, which is a naturally competent bacterium here, right? So let's also look at the process by which you can make non-competent cells competent. And there are two such processes that are most commonly used. So first, of course, is to give a chemical treatment, which means that, you know, the cells are chemically treated, and most commonly they're treated with calcium chloride at the concentration of 50 millimoles per liter. And the cells that are taken for this process are actually have to be in log phase, or the actively growing phase of the bacterial growth curve. And then, of course, these cells can, once treated with calcium chloride, they become more permeable to foreign DNA. And when they're given a heat shock where the temperature is suddenly changed from very cold, around 0 degrees centigrade, to extremely high temperature of 37 to 42, the cells are able to take up foreign DNA very easily. Right? So this is basically your one method of doing it. Then, of course, you could actually treat the cells with electrical shocks, and that also changes the property of the cells and makes them more amenable to accepting foreign DNA. So cells may also become competent when treated with brief pulses of electricity. Such cells undergo transformation by electrical shock. So here is your two methods. You can either use chemical method by treatment with calcium chloride, or you could give an electrical shock. In both cases, the cells, the outer membrane of the cell would become more porous, and resulting in acceptance of the foreign DNA much easily as compared to a cell that is non-competent. And then, of course, you could add your recombinant vector now. Once the cell is in the competent state, you can add a recombinant vector and give either a heat shock or a electrical shock again, which will allow the DNA to move into the host cell. And then, of course, over time, the competence is lost and the cells regain their normal membranes. And that is the process of recovery, during which time the cells repair the damaged cell membrane or cell wall and become non-competent again. So this is the process of cell recovery, where now the cell has regained its outer covering, which was either the cell wall or the cell membrane. So competence per se is not a permanent state. It is a transition state, and we take advantage of this transition state to ensure that our vector is able to enter into the host cell. Right? So this is your competent cells. So once the cell reach the competent state, they can be transformed, and there are several methods of transformation. These include heat shock, electroporation, biolistics or gene gun, protoplast fusion, and microinjection, and we'll discuss each of them as we move along into the lecture. But also, I want you to look into the very important classical experiment of Frederick Griffith, which is also called as transformation experiment. And also, I wanted to look into the significance of the experiment. These are some of the basic experiments in microbiology to establish that DNA is the genetic material. So the most common method of transformation is heat shock. And as the name indicates, there is a sudden change in temperature that sends the cells into shock. And during this time, the plasmid is able to enter into the host cell. So these are our competent cells treated with calcium chloride. And you have the recombinant vector alongside. Alongside, you also have self-ligated vector, which I'm not showing here. So this ligation mix is incubated along with competent cells at 0 degrees centigrade for around 30 minutes. And then, of course, they are suddenly given a transient exposure to high temperature, 2 minutes at 37 to 42 degrees centigrade, depending on the strain of the host cell that is being used. When they are suddenly transferred from 0 degrees centigrade to a very high temperature of 37 to 42, the cells undergo a shock. And during this time, the plasmid vector, which carries your DNA of interest, is able to insert into the host cell. Right? So this is one of the methods of transformation, the most common. The simplest method does not require very sophisticated instruments. So upon incubation at 0 degrees centigrade for close to 1 hour with the recombinant vector, and then suddenly transferring it to high temperature of 37 to 42 degrees centigrade, the cells are able to very easily accept DNA, foreign DNA, and what you get is a transformed cell. This is again in the competent state, and then of course over time it becomes non-competent because the whatever biochemical changes that occurred in the cell membrane or cell wall are repaired, and finally you have the host cell that carries your recombinant vector is what we prefer, but you also get host cells that are transformants that carry the self-ligated plasmid. So another important method of transformation is protoplast fusion. So this is your host cell, and the host cell is treated with enzymes so as to dissolve the outer cell wall. That is basically to expose the protoplast. This because it does not have an outer covering anymore. It is more amenable to taking up foreign DNA because there is no first line of defense or an outer limitation boundary that prevents free diffusion or free entry of substances from outside. So enzymes that degrade yeast, fungal, and plant cell walls are used to remove the cell wall, and intact protoplast can be obtained. Protoplasts generally take up DNA quite readily. However, you can also supplement with electroporation, where you give an electrical shock, which makes the cell even more porous and readily accept foreign DNA. After transformation, the protoplasts are washed to remove the degregative enzymes, and the cell wall is spontaneously developed again. So here is a protoplast. This protoplast, when you add your recombinant vector, it is able to take up the recombinant vector very easily and very comfortably. And then, of course, you wash the protoplast so as to remove the enzymes that are required for digestion of the cell wall. Once those enzymes are removed, you have the restoration of the cell wall, and finally you have a cell that has been successfully transformed, the recombinant vector. Right? So this is another protoplast fusion is one of the important techniques, more so in plants for transformation. Then you have another method of using shear force to force a plasmid into the host cell or a vector into the host cell. So this is known as biolistics or gene gun. You basically coat your DNA, your vector onto a nanoparticle, and basically projectile it with such speed, high speed, that it is able to pierce through the host external wall and membranes and enter directly into the host cytoplasm. And this can also be used for targeting organelles and this which include plastids and mitochondria and so on and so forth. So let's have a look at how this works out. This is your vector. This vector is basically coated onto a nanoparticle, and that is then projectile into the cell with such force that it is able to break open the point of at the point of contact the host cell. It is able to break open the membrane or the cell wall and is able to insert into the cytoplasm. And this can also be directed into host cell organelles like mitochondria and chloroplast. So gene gun or biolistics. A gene gun or biolistics is a micro projectile delivery system. So this is a projectile delivery system at the micro scale. The projectile is an elemental particle with heavy metal coated with a payload of DNA. And typically what you have is a plasmid here. And this can also target organelles, including plastids and mitochondria. And in the activity column, I want you to name a template independent DNA polymerase. And I'm sure you would be remembering it from the previous lectures. If you don't, then of course, again, you need to go through the previous lectures before you come to this lecture for more details. So this is your biolistics or gene gun. Another important method of transformation is microinjection. Microinjection is a direct manual method of introducing the foreign DNA into the host cell. So here you are. This is your recombinant DNA, which is present in a micro syringe. And then you directly pierce into the host cell and place your syringe in a position where you can directly deliver the piece of DNA into the host cell here, right? And the positioning of the needle is helped by what is known as a micromanipulator. And then, of course, each cell is individually injected under the microscope. And this is a manual, non-automated, time-taking process, but then with expert hands, the efficiency may be very high. So microinjection is a direct manual method of transformation, introduces DNA into either the cytoplasm or the nucleus. It is a microsurgical procedure using a glass microcapillary pipette, a precision positioning device, which is known as a micromanipulator to control the movement of the micropipette, and a microinjector. And as I already told you, each cell has to be individually injected. And of course, it is manual and it takes a lot of time. But then, of course, it may also, depending on the expertise level and the skills of the person who's doing the microinjection, the results may be much more efficient. Some of the key terms that you have to remember here include microinjection, microcapillary, micromanipulator, and of course transformation. In the end, I discuss electroporation. Electroporation is a very common technique of transformation of host cells with a recombinant vector or a self-ligated vector. The name is very indicative of what is done here. So you are given, the cells are given brief pulses of electricity, and during the time the cells are exposed to brief pulses of electricity, the negatively charged DNA is able to enter into the host cell. Electroporation was shown to work way back in 1988. And today, along with heat shock, these are the two most common methods of transformation that are used in research labs. You have a competent cell here, you have a recombinant vector along with, so basically this is a ligation mix. So you have a recombinant vector, also you may have a self-ligated vector, and then you give a electric pulse. And during the time of the exposure to electric pulse, the cells become receptive and allow negatively charged DNA to move into the host cell, finally resulting in a transformed cell here. So after the cells have undergone transformation, the cells are put into the phase known as recovery phase. During the recovery phase, the cells are given rich nutrient medium so that they're able to grow fast and are able to recover from the shock that comes along with transformation. So this is basically your recovery phase here. In the recovery phase, the cells are transformed in an antibiotic-free, rich liquid medium, which is known as the SOC medium, or the full form for SOC medium is super optimal broth with catabolite repression medium. And then, of course, if you see, this is the transformed cells are cultured in 1 ml of pre-warmed SOC medium at 37 degrees centigrade with shaking at 225 RPM for 1 hour. The composition of SOC medium is here. It consists of 2% tryptone, 0.5% yeast extract, 10 millimolar NaCl, 2.5 millimolar KCl, 10 millimolar MgCl2, 10 millimolar MgSO4, and 20 millimolar of glucose. So this is the composition. So some of the key terms that you must know here, and I'm sure you would have been taught in your molecular biology classes as well as in microbiology classes is number one, the lack of operon. Lack operon, so operon is basically a structural and functional organization of genes of the same pathway in prokaryotes. So the genes of the same pathway in prokaryotes are both structurally and functionally linked into one single group known as an operon. All these genes of the same pathway are placed next to each other. They form a polycystonic mRNA, and also they are either expressed all of them or none of them. Right. So this is your lack operon. Of course, you can go into the details of this in your microbiology and microbiology classes. One of the key genes in lack operon is the beta-galactosidase gene, and this gene is very important also in terms of screening. And we'll see this when we talk of vectors. We'll see how the color-based screening, which is the blue-white screening, is done with blue-white selection, is done on the basis of beta-galactosidase getting functional or dysfunctional. And then, of course, the catabolite repression is also a term that is associated with the operon functioning and refers to the fact that as long as the glucose is present in the medium, the lack operon is not switched on. And when the glucose is going down, the concentration of glucose is going down and comes below a critical level, the lack operon is switched on so that now lactose can be metabolized and you can synthesize glucose and galactose, right? So this is your lack operon. So let's do a quick revision of the steps in cell-based cloning before moving to selection. So of course you have your insert, which may be blunt end and has to be converted to sticky ends. Then you have your vector, which is usually a closed circular DNA, which has to be opened by restriction enzymes. And then the two are put together, and ligase allows for the formation of a recombinant vector, where your insert now is well within the vector that you're using here. And then this vector has to be inserted into the host cell by the process of transformation. And transformation can take place by heat shock, electroporation, gene gun, or protoplast fusion, microinjection, etc. And then, of course, you get the transformed cell here, where your recombinant plasmid or your non-recombinant plasmid gets into the host cell. And you also have a group of cells that are non-transformants, which do not have any of the properties conferred by the vector here, right? Then, of course, the cells go through a recovery phase so that they come out of the shock during transformation. And once that is done, the cells are now subjected to selection. And there are several methods of selection that are used. In this case, in the lecture today, we would be checking into inactivation of resistance as one of the methods. And then as we move into the lectures on vectors, we will look into the other methods of selection. One of the most common methods of selection is what is known as a color-based selection, where you have two types of colonies, blue and white. The white colonies are recombinants and blue are your non-recombinants. But this color-based selection will study as we move into the vectors. For now, we focus our attention to inactivation of resistance as a method of selection for recombinants. So here you are. You have your recombinant plasmid, you have a non-recombinant plasmid. This is a ligation mix. You have your host cell, which is specially treated and made competent. You either go for electroporation or heat shock most commonly, but you can also go for protoplast fusion, or you can also go for microinjection, or you could go for gene gun or projectile. In any case, whatever method of transformation you select, finally you get three populations of cells. So this one here, where a recombinant plasmid has got into the host cell, this is known as a recombinant. Then this one here, which is where the self-ligated plasmid has gotten into the host cell. So this is your transformant, but it is not a recombinant. And then you'll have cells where the plasmid does not get into the host cell. So these are your non-transformants. You have three populations of cells: recombinants, non-recombinants, and non-transformants. Now, the method to select for the recombinant cells is inbuilt into the vector. So there are mechanisms by which you can, depending on which vector you're using, there are a specific method by which you will be able to identify which of the cells are recombinants and which are not, right? There are two most common methods of selection. One, of course, is resistance inactivation, which we're going to study right now. And of course, the other important one is blue-white screening, which I've given you a brief, but we'll do this in more details with actually what is happening at the molecular level when we do the vectors. So now let's have a look at the simplest selection method, that is the resistance inactivation in plasmid. So the most common method of, the most simple method of selection is the resistance inactivation in plasmid, which you're going to see in the plasmid vector, that is PBr322. Before going into the details of selection method, there is one important technique that you need to know, that is known as replica plating, or how to copy a cell colony into another plate, right? So this is what is replica plating here. So as the name indicates, replica plating essentially means that you're making a copy of a previous plate. So here is your plate. This is called the master plate, which is the original copy of the plate. And what you want to do is to make a photocopy of this so that the colony positions on this plate and the second plate correspond, right? So what you do here is, this is your incubated plate where you can see colonies of your bacteria coming up here. So what you do then is to use this prop. This is of the shape of a circular disc with a handle on the top. So this prop is covered with a muslin cloth, and this prop can enter very smoothly into the petri plate and touch the surface of this master plate here. So this is what you do. You cover it with a muslin cloth, and then you basically push it into the master plate. Some of the colonies that were there on the master plate some of the cells in these colonies would now get transferred onto this, onto the muslin cloth at exactly the same position where they were in the master plate. And now what you do is you make a replica plate by taking another plate, which the culture medium, and onto this you basically imprint the prop here. So this is what you do here. So this is a replica plate, and you take the prop and allow it to touch the surface of this culture plate here. So corresponding positions of each colonies can be marked here, and they are transferred. And then, of course, when you incubate this replica plate for overnight, you will have the growth of colonies on this replica plate, and this will be actually exactly at the same positions as it was in the master plate. So effectively, what you've done is you made a photocopy of the original plate here. So this is a technique called replica plating, and it is commonly used in screening, particularly in case of resistance-based selection of recombinant cells. And we'll see how this is used in this particular context. So let's move on, and let's see another important point in vectors is the presence of resistance genes here. So you have I show you an example of PBr322, and in PBr322 there are two resistance genes. One that is known as ampicillin resistance gene, and the other that is known as tetracycline resistance gene. So when this PBr322 enters a host cell, because it has the properties of tetracycline resistance and ampicillin resistance, the host cell also becomes tetracycline and ampicillin resistance. However, in PBr322, you also have restriction enzymes, and these restriction enzymes allow you to insert your DNA of interest either into the tetracycline resistance gene or into the ampicillin resistance gene, right? So here you are. If you see, so the DNA of interest or insert may be inserted into one of the two resistance genes, any one of the resistance genes here. Let's assume that it inserts into the tetracycline resistance gene. So let's now assume that when you do your ligation, the insert is coming and sitting into the tetracycline resistance gene, which means now that the tetracycline resistance gene is disrupted, which also essentially means that the cells that take up this recombinant plasmid will be sensitive to tetracycline. Right? So here you are again. This implies that the tetracycline resistance gene will be disrupted. Such cells will be tetracycline sensitive, and we'll use this as our indicator or for the identification of your recombinant transformants. So here you are. So coming back to the process again, so let's say plasmid PBr322 confers resistance against ampicillin and tetracycline. So we know that. We now assume that the insert ligates within the tetracycline resistance gene. So when the insert is into the tetracycline resistance gene, the tetracycline resistance gene is now disrupted and is non-functional. That results in loss of resistance to tetracycline antibiotic, right? So here you are, your recombinant plasmid, non-recombinant plasmid, your host cell, your process of transformation, either electroporation or heat shock, and the resulting three cells here. So this one is a recombinant, this one is a non-recombinant, but transformant, and this one is a non-transformant because there is no plasmid that has entered into this host cell. So these are your classes. Now, if you look at the sensitivity of or the sensitivity to antibiotics for these cells, so let's start with the bottom one here, which is a non-transformant. Because there is no plasmid here, any property that is conferred by the plasmid is also not present in these cells. So therefore, these cells will be tetracycline sensitive as well as ampicillin sensitive, right? So this is a cell that is both tetracycline sensitive and ampicillin sensitive. This is a non-transformant. Coming back to the second one here, if you see, this cell here, where the self-ligated plasmid has come in, which means that both the tetracycline resistance gene and the ampicillin resistance gene are absolutely intact and active. So therefore, these cells would be resistant to both tetracycline and ampicillin. Coming to the recombinant now, you have your recombinant cell here, and in this case, you know that the insert or the DNA of interest has come and disrupted into the tetracycline resistance gene, which means these cells would be sensitive to tetracycline but resistant to ampicillin, right? So this is the functional nature of the three categories of cells that you get. Recombinants are tetracycline sensitive and ampicillin resistant. Non-recombinants are resistant to both tetracycline and ampicillin, and non-transformants are sensitive to both tetracycline and ampicillin, right? So this is the nature, and this basic nature would be used for screening of the cells here, for selection of the cells that are recombinants. So let's move on and see how this works out. So transformants are ampicillin resistant, and non-transformants are ampicillin sensitive. So the first important point that you can take back from here are that transformants are ampicillin resistant. These are transformants, and both are ampicillin resistant, while non-transformants are ampicillin sensitive. So the first round of selection happens by re-inoculating these cells into a Petri plate that contains ampicillin, right? So ampicillin, when you do a selection here, so these non-transformants will not be able to grow, and both the transformants, which is either the recombinants or non-recombinants, will be able to grow in those plates here. So this is your color coding for now. So this is your non-transformant here. Then this is your non-recombinant transformant, and this is your recombinant transformant here, right? So if you look at the sensitivity here, so these are your non-transformants, tetracycline sensitive, ampicillin sensitive. These are your non-recombinants, which are tetracycline resistant, but and also ampicillin resistant. And then you have the recombinants, where the DNA of interest has come and inserted into the tetracycline gene, resulting in loss of tetracycline resistance. So these are tetracycline sensitive, but they also have the ampicillin gene, which is intact. So these are ampicillin resistant. Now what you do is you basically do you first inoculate this mix onto an ampicillin plate. So now you know that your non-transformants are ampicillin sensitive, which means they will not be able to grow here. So only two types of cells can grow here, which are the transformants. And amongst the transformants, you have the non-recombinants and you have the recombinants. So if you see here, when you inoculate and you incubate for some time, after some time, you'll be able to see two types of colonies here. You have the green colonies, which are your recombinants, and you have the blue colonies, which are your non-recombinants. And this is not actually, the color would be exactly the same. I'm showing you for the purpose of discriminating between the two. I'm showing the recombinants as green and the non-recombinants in blue. They would all be of the same color. Please remember that they would all be of the same color here. And then what you do is replica plating. In this case, only the transformants are able to survive. The non-transformants do not survive. Then next you do is replica plating. And in replica plating, what you do is, in the second plate, you also add, along with ampicillin, you add tetracycline as well. So you know these cells, the recombinants, are tetracycline sensitive. So when you do a replica plating from this plate onto this plate, and in this plate, you also add ampicillin and tetracycline, only the non-recombinants are able to grow. The recombinants will not grow. So the only colonies that you'll find here will be of blue type and not the green type. And again, once again, to emphasize, the color of the colonies is the same. Just to discriminate between the two types, I call them blue color and this one as green color. So now what you do is after you've done your inoculation and incubation, you'll get a certain set of colonies. These are basically coming from the cells that are having the self-ligated plasmid here, right? And then what you can do is you can compare the two plates. The cells that are present exclusively in one plate and not in the other represent your recombinant colonies here. So now you can pick up these colonies and take them forward for further culturing or for making the glycerol stock. So this is your ampicillin and tetracycline plates. Ampicillin and tetracycline both are present, and only the transformants with self-ligated plasmid are able to grow here and not the recombinants, because in recombinants, the tetracycline resistance has been disrupted due to the insertion of the DNA of interest here. So this is what you get. So colonies growing in AMP plus plates, but not in AMP plus and TET plus plates, carry the recombinant vector. So this is your resistance-based selection. You have your master plate, which has a certain number of bacterial colonies here. And then, of course, you do a replica plating. The master plate is grown in ampicillin alone, where the non-transformants will not grow. And during the replica plating, this plate also contains ampicillin as well as tetracycline. And therefore, the recombinants, the recombinant colonies will not grow here. And therefore, what you can do is the colonies exclusive to one of the two plates represent the recombinant, which is shown in green color here, although the color would be the same for all the colonies. So if effectively you're looking in this plate, the colonies that have not grown in this plate are the ones that are recombinant. And because the positions are the same, you can refer back here, and these colonies that are present here but not in these positions on the replica plate are the actual recombinant colonies. So the colonies absent in the replica plate but present in the master plate represent the recombinants and may be picked up and inoculated in a fresh plate or grown in liquid medium and stored as glycerol stocks for later use. So resistance-based selection is a very simple method of identifying your recombinants. However, it is slightly cumbersome in the sense that you have to inoculate twice and you have to incubate twice. And during replica plating, if there are mistakes, then of course you will not be able to identify your actual recombinants. So what people have now done is they have moved on to one-step method of distinguishing the recombinants from non-recombinants, and that is based on the color. So this is what is known as color-based selection. Here, the colors of the colonies are true. So the most common type of color-based selection is the blue-white selection, where you have two types of distinct colonies on the plate, one that are blue in color and the other that are white in color. And in this case, white colonies represent recombinants and the blue colonies represent non-recombinants. So white colonies are the ones where your recombinant DNA is present. The blue colonies are the ones where the non-recombinant DNA or the self-ligated plasmid is present. And the selection here is based on the capability of the cells to be able to synthesize a functional beta-galactosidase. A functional beta-galactosidase is not formed in the white colonies, while it is formed in the blue colonies. And so I'll be discussing this in more details as we move into vectors in the next lecture. Then let's move on